Okay, welcome to the next video for sensation and perception. This is on vision. We're going to start with the parts of the eye, and then we'll get into how vision is actually working, okay? So let's start with the different parts of the eye. We're going to start from the outside and work its way in. This picture is going to be a side view, so as if we like cut your eyeball in half and we're looking at the inside of it, okay? So let's start on the outside and work our way in. The very first section that we want to talk about is the cornea. The cornea is the transparent covering of your eye. Think of it as just kind of a shield. It's there to protect the inside of your eye. It's meant to, keep out dust and all of that kind of thing. But if you scratch your cornea, it's probably pretty painful. The pupil is the next part we're going to talk about. The pupil is the black part of your eye that you can look at. This is actually an opening. This is where light enters into your eye. So this is just a hole. That's all it is. It's the black part, but it's a hole. And the pupil, we talk about the pupil constricting or dilating, and that is controlled by the iris, which is the colored part of your eye. This is the muscle that allows the light into your eye. So our iris actually makes our pupil opening smaller or larger depending on the amount of light that there is in a room. If it's a very bright room, your pupils will get really small because it doesn't need to bring in a ton of light. But if it's a dark room, your pupils will get really wide because it needs more light to enter in, okay? The next part is the lens, which is right here. We're going to talk about, think of this just as you would like glasses that you put on. Your lens of your glasses are going to focus your vision, right? And that's exactly what your lens does. It focuses light onto your retina. The lens also has a special duty is that it flips the image upside down. So when we take an image into our eye, it gets flipped upside down onto our retina, and then we see the retina then gets that information to our brain. The next part is the actual retina. The retina is the back of the eye here. Think of this as like your projector screen. The lens is going to project that image onto the retina. This is where all of your visual sensory receptors are, and those are rods and cones. We're going to talk about those in a minute. But this is also where transduction happens. Transduction is an important word, so I want you to write this down if you haven't already. Transduction is the idea of when we're taking a sensory stimuli out, external stimuli, so in this case light, and our body is going to transduce it or transfer it to neural messages, okay? So we are now transforming that light into a neural message, and this happens on that retina. Part of the retina is called the fovea, which is back here. It's this little bump here. This is the central point of focus. This is where most of our eye's cones are. They're all kind of clumped around that fovea, and we'll talk about what that means in a little bit. But this is the fovea here. The next part is our optic nerve. That's this yellow piece here. The optic nerve is physically what is connecting your eyeball to your brain, okay? So the information gets transduced into a neural message, which gets sent via the optic nerve, first to the thalamus, which is our sensory control center, then to our occipital lobe, which has our visual cortex, which processes the information that we're getting. Finally, we have our blind spot. The blind spot is where the optic nerve connects to our eyeball, okay? And there are no receptors here because it needs to connect somehow. So there are no, you can't see in that area. We're going to test your blind spot, but what's really crazy is that our brain actually just fills in our blind spot for us, and it fills in with whatever it's surrounded by, because our brain doesn't like to have holes in the information. We'll test our blind spot in class, but it's pretty cool. Okay, so let's talk about how we actually perceive images, right? So when we think of light, you can think of them in terms of waves. I like to think of them in terms of just like waves in a wave pool, okay? So hue, or the color of the light coming in, is going to be based on the wavelength, okay? The wavelength is the distance from one peak of a wave to another, okay? So if you're in a wave pool, it would be measuring from the top of one wave to the top of another wave, okay? That's wavelength. Shorter wavelengths are going to be bluish colors. Longer wavelengths are going to be reddish colors, okay? And this is a better visual to kind of show you. Here's our short wavelengths over here. We got those violets and bluish colors. And then we have our long wavelengths over here, which are going to be reddish colors. So that's how we perceive different colors. The intensity or the brightness of the light is determined by the amplitude of the wave. So how tall those waves are. So is it a 6-foot wave in the wave pool or is it an inch wave? Okay. If it's a 6-foot wave, we have more energy, right? There's more water. And so that would be brighter. If we have low amplitude or a short wave, that's going to be a dull color because there's not as much energy in it. Okay, let's talk about the two different types of sightedness that people can have. So normal vision, our lens focuses a image on the retina here. But if you are nearsighted, you, which is also called myopia, which is important for you to know, if you are nearsightedness, you can see close-up objects easily, right? That's no problem. But it's those far away objects that you have issues with. That's because of the shape of the eye. The eye in someone who is nearsighted, like me, is that you have this elongated eye, right? So here's the typical eye, and then in a nearsighted eye, you have it's elongated. And what's happening is that the image is actually focusing before it hits the retina. So there's no real good, you can't see it very well. So this is an example of how it actually looks, okay? So here you can see close up pretty easily, but then those far away things are difficult to see and compared to normal vision over here. Okay, farsightedness then is the opposite. You can see far away objects pretty clearly, but then those near objects are out of focus, and that's because our eye is shortened. This is also called hyperopia. Okay, so this is the shape of a far-sighted eye. Again, here's a typical eye. Here's the far-sighted eye. It is taller than it is wide. Okay, and what's happening is that the image that's being projected onto the retina isn't focusing till behind the eye. Okay, and so you can't see those things that are close up very easily. Here's an example over here. Here's the faraway objects that are pretty clear. Then the nearby objects are blurry compared to normal vision here. Okay, so let's actually talk about the retina because that seems to be where everything is happening, right? So then retina is made up of, it's the inner surface of our eye, and it's made up of cones and rods, okay? Rods are going to be, actually let's talk about cones first. Cones are going to be color, okay? They help you see color. Cones, color. They also help you pick out fine details, okay? Rods are going to do, they're more light sensitive, and so they're going to do, you know, black and white night vision kind of things. But cones are going to be your color. Our retina is also made up of layers of other neurons, ganglion cells and bipolar cells. These process the visual information, okay, and then they send them to the brain. So what happens when we see something? The light enters through our eye, through our pupil. It goes through the lens, gets flipped upside down, and goes to the very back of the retina. So it comes in through here and gets shot all the way to the back of the retina here. That's where the cones and rods are located, those photoreceptor cells, okay? Then they get sent up a little bit forward to the bipolar cell and then to the ganglion cell, which then sends the message to the brain, okay? So it goes all the way to the back first, then goes up to the bipolar cell, ganglion cell, to the brain, okay? So that's how it works. In the fovea, which remember is the central point of focus in the retina, this is where all of your cones are located, which makes sense. Where we can see fine details, where we can see a lot of color. You can see a ton of detail and color when you're looking directly at something, right? It's your peripheral vision that you can't see a lot of detail and color. That's because all of your cones are in or right around your fovea, and then your rods are on the outsides of it, okay? These are your color or your light vision, okay? So think about it. Why is your pupil dilating? or why is it contracting? This is an important question, okay? If you can understand this, you can understand a lot. So when our pupil dilates or gets larger when we're in a dark room, it's because we need to expose more of these rods, right? We have more light coming here. We need those rods to be able to determine the information that's around us. So when our pupil dilates, it gets larger to let more light in and expose more rods, okay? This also allows us to see a little bit less color, right? Think about your night vision. When you're seeing things in the dark, you're not seeing great color, but you can see the images, okay? So photoreceptors, like we said, rods are sensitive to light. Cones are sensitive to color and fine detail. In this image, we can see here that our these right here are the cones, okay? We see the cone shape. Cones connect directly to one bipolar cell and then to a ganglion cell. Why do you think that is? Well, because we can see more detail with them. So we can get a lot more information from this one cone than directly to a bipolar and directly to a ganglion cell. Rods, however, right? Rods, there's about 3 rods per one bipolar cell. And so you're not getting quite as much detail from those rods because they have to share one bipolar cell. Okay, that's pretty important right there. Okay, let's look at those specifically. So bipolar cells receive the message from the photoreceptors, the rods or the cones, and then they transmit them to the ganglion cells. The ganglion cells form the optic nerve. Okay, and the optic nerve, remember, sends the messages directly to the brain. Cones each have their own bipolar cell to get more information. Rods share bipolar cell. There's about 3 rods per bipolar cell. Okay, we just said this. This is a little bit of a review. We're just like looking at it more than once. Okay, finally, let's talk about feature detectors. Feature detectors are specific cells in our visual cortex that respond to features such as edges, angle, length, and movement. Okay, so we have specific cells that just look for the edges, just look for the angles of things, just look for the length of things, just look for the movement. These are called feature detectors. There's also feature detectors that are specific to the human face. If you remember back to the bio unit when we would split the brain, they're probably located in the right occipital lobe, right? Because that's the side of our brain that recognizes faces. So these are feature detectors. Think of these as specialized cells for certain images. The cool thing about feature detectors, though, is that we use these feature detectors simultaneously. So this is called parallel processing. The brain divides information up into color, depth, form, and movement, but we process all of that information at the same time. So when you're looking at an object, you're not just seeing the edge first, then the color, then the movement, right? You're doing it all at once. That's called parallel processing because all of those things are happening in tandem with each other.